Okay, so there, we're recording now. So this recording will be in two parts then. I didn't see, normally when I record, you get this thing here that tells you recording. That's why. That's why I got nervous. All right. So if you go from nitrate to nitrite to nitrogen, that's nitrogen reduction, nitrate reduction. Denitrification is ecologically the main way you lose nitrogen from soil. And then an exquisitely oxygen sensitive process is sulfate reduction, where you take sulfate minus, come on, you, -2. And you reduce this either to sulfite, but more likely you'll reduce it to elemental sulfur. Or in some cases, if you reduce it all the way, you reduce it to sulfide, which is sulfur with a -2 charge. That's sulfate reduction. Sulfate reduction is exquisitely oxygen sensitive. Those are examples of anaerobic respiration. Nitrate reduction to either nitrite or nitrogen gas, or sulfate reduction, SO4 -2, to you reduce that to either elemental sulfur or to sulfide. And this really cool picture here shows you the carriers embedded in the membrane. You pump the protons into that periplasmic space. And now to get those protons back into the cell cytosol, you're going to use this enzyme called the ATP synthase. The ATP synthase. So the ATP synthase is a very complicated protein. It has a pore that will admit protons back into the cytosol. Linked to that is a set of proteins that synthesize ATP from ADP and phosphate. The energy for this synthesis comes in the form of readmitting these protons back into the cytosol. So this readmission here, essentially, of these protons drives the synthesis of ATP. This process whereby you use a proton motive force generated by electron transport chain to generate ATP, that is called either cellular respiration or oxidative phosphorylation. Oxidative phosphorylation, let me oxidative phosphorylation, otherwise known as cellular respiration. You use a proton motive force to generate ATP. That proton motive force is generated by an electron transport chain. You generate that electron transport chain, use that to make ATP. So the membrane, as we see here, your internet connection is unstable. Let's try that. You have your carriers they pump the protons out here. In order to readmit those protons back into the cytosol, the cell has this thing called an ATP synthase. The ATP synthase takes these, readmits them, but if you readmit them, you pay a toll. That toll is the synthesis. You take ADP, an inorganic phosphate, and you use that to make ATP. So in mitochondria, it seems as though for one NADH, you're going to make somewhere around 2.5 ATP. That's kind of the way the numbers fall out for now. For every reduced flavine, you're going to make about 1.5 ATP. Now, some books will say it's 3. It's probably 3 in chloroplasts, not in mitochondria. In bacteria, the best numbers available say it's about this number here, about 2.5 per NADH. It's about 1.5, maybe it's 1.46 or something like that, or 1.51 or something. Depending on the papers, the numbers change, but that's a good ballpark. So from this, let's do a little back of the napkin calculation now. So per glucose, okay, so glucose, how many ATP do you make per glucose? How many ATP do you make per glycolysis per glucose? How many do you make?
4.
You make 4 minus 2 or 2. And how many NADHs you make? Okay. All right, then there's the PDH catalyzed reaction. How many ATP do you make? Don't make any, do you? But you make a couple of these, don't you? Krebs cycle. How many of these you make per glucose? 3 times 2 or, okay. And we didn't include our flavines over here. How many of those do you make per glucose? 2. How many ATP do you make? 2. Make 1 per turn of the cycle. All right, so that's 10 NADHs, 2 FADH2s, 4 ATP net. You're going to make, so that's going to be what, 3, right? This is going to be 25, right? And if my math is correct, that's 32. You make 32 ATP per glucose. That's about how much energy you're going to get per glucose. Right. 2 plus 2 plus 6 is 10, right? Did I do that right? Okay, so we sum that up. Plus, so we get 32 ATP per glucose total. That's if you use oxygen as your terminal electron acceptor. If you're an organism capable of anaerobic respiration and you use nitrate, the amount of ATP you get per glucose will be less. Instead of 32, it'll be more like 21, 22, something like that. If you use sulfate, it'll be more like 12 to 15. So oxygen is the best electron acceptor you can have. Others will function, but they don't work as well. All right. So the carriers for the electron transport chain are embedded in the inner mitochondrial membrane of our cells and other eukaryotes. We talked about this, oxidative phosphorylation. You're going to reduce molecular oxygen to make water. And sometimes the electron transport chain goes awry. And instead of making water, it'll make things like superoxides, radicals, or it'll make hydrogen peroxide. These can be detoxified using this 1-2 punch set of enzymes we talked about before, catalase and superoxide dismutase. Anaerobes will tend to, obligate anaerobes will tend to lack these enzymes. Aerobes will tend to have these enzymes. We will do a test for catalase in the lab. It's very easy to do. You take a slurry of cells and you put them in a drop of hydrogen peroxide. Catalase takes hydrogen peroxide and it makes oxygen out of it. So if there's catalase, the cells will go gurgle, 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 and they'll make lots of bubbles. You don't see any bubbles, your catalase negative. You see lots of bubbles, your catalase positive. Very easy test to do. For anaerobic respiration, if you use nitrate, then instead of cytochrome oxidase as your last enzyme in the electron transport chain, you'll have nitrate reductase. Nitrate reductase. There's an entire gene expression cascade that detects the presence of oxygen that keeps nitrate reductase repressed. When there's no oxygen, that cascade now activates the expression of nitrate reductase and suppresses the expression of cytochrome oxidase in anaerobic and facultative anaerobic bacteria. Nitrate reductase can reduce nitrate to nitrogen gas. Nitrogen gas will be released from the soil. That's called denitrification. And if you don't have an electron transport chain, if you just make your ATP with glycolysis, then you carry out a fermentation. Please, in fact, one of the questions on your self-test is, fermentation means you make ethanol. No. Fermentation is ethanol is one of the ways you can ferment. Fermentation, ethanol is a possible product of fermentation. So fermentation can produce reduced organic compounds, of which ethanol is one example. You yield small amounts of ATP and you tend to grow more slowly. Many organisms that are aerobic in the air, when there's no air, will revert to fermentation. Some organisms are permanent fermenters. One of the best places where you see that is in the rumen of a cow. Inside the cow, what do cows eat? Grass. What's grass filled with? Cellulose. Cows have lots of microbes that degrade cellulose to sugar. Many of these organisms then ferment the sugar to organic acids, and then these other organisms take the organic acids and convert them to carbon dioxide, and then methanogens convert the carbon dioxide and hydrogen gas to methane. And that's why cows belch, because they're belching out methane. And that's why people concerned about the environment are concerned about cattle, because methane is a greenhouse gas. How much methane, a bunch of dairy cows produce is debatable. But this has led to efforts essentially to make cultured meat and stuff like that and what have you that don't use cows. So here are two examples. We have an ethanol fermentation where you take pyruvate and decarboxylate it using a pyruvate decarboxylase enzyme. And then that acetaldehyde is reduced by aldehyde dehydrogenase to form ethanol. The other option is you use lactate dehydrogenase to reduce pyruvate directly to lactic acid. These are two possible fermentations. This fermentation over here, as you can see, produces carbon dioxide. This one does not. The products of fermentation sometimes are important industrially. So ethanol, butanol, acetate for making vinegar, vitamins, antibiotics, hormones, many of those are made by organisms that are co-opted by companies to make some of these compounds. And microbes become the slaves, if you will, of these companies to make these compounds. Fermentation is not just for making ethanol. So underneath many of the trees in our forest behind the campus, those leaves are being degraded back to more basic components. They're being recycled by decomposers. Many of those decomposers are fermenters. So fermentation is a process that is very important ecologically speaking. And it's also important inside our GI tracts. We have lots of fermentation going on in our GI tracts right now. The bagels or the pancakes or maybe the eggs you had for breakfast. Some of the indigestible material is now being digested by microbes in your GI tract. And they're making short chain volatile fatty acids. Many of those are very good for you. Some of those, in fact, may prevent the onset of Alzheimer's. So fermentation is something, I heard a guy behind a pulpit once, he was a visiting guy, he said, there was no fermentation in the Garden of Eden. Fermentation happened after the fall. And I sat there going, wait a minute. And so my roommate said I cornered him. I don't think I cornered him, but I asked him after the sermon, I said, well, what about leaves? What about short chain fatty acids? What about vitamin B12 vitamin production in your GI tract? What about that? That's all? Oh, we could make our own B12, and then after the fall, we could. Really? Are you sure about that? And he got quite mad at me and then stormed off. I was calm, really. I was calm. That makes no biological sense at all. And it makes no theological sense if Eden was this self-sustaining place either. What's an organism to do with non-carbohydrate compounds? Let's go back to our makeshift table here. Here's our blackboard. So we have glycolysis. Glycolysis feeds to the Krebs cycle, which then feeds both of these feed to the electron transport chain, which makes ATP. That's for glucose. What do we do with proteins? Proteins are polymers of what? What do you link together to make proteins? They end with acids. Thank you, amino acids. So you're going to use proteases to degrade the proteins to amino acids. Several of these amino acids either feed directly into glycolysis, or many of them feed into the Krebs cycle, which will then produce reductant that goes to the electron transport chain and makes ATP. What about lipids? What are lipids made of? They're fats. What are fats made of? They're made of glycerol, esterified to fatty acids. You're going to break this down with an enzyme called a lipase. You get free fatty acids and glycerol. Glycerol feeds straight into glycolysis. The fatty acids are broken down by a process called beta oxidation. They're broken down to acetyl coenzyme A. Acetyl coenzyme A feeds straight into the Krebs cycle. So metabolically, all roads kind of lead here. In our cells, beta oxidation occurs inside the mitochondria. In bacteria, it occurs in the cytosol. So let's rehearse. Proteins are degraded by 2 amino acids feed into glycolysis, or they get degraded to carbon dioxide and high energy electrons. Those high energy electrons go to make very, very good. Lipids are degraded by lipases to glycerol and free fatty acids. Glycerol feeds directly into, and then the fatty acids are degraded by, say it, beta oxidation. Say it again, beta oxidation. 2, that feeds into in the house that Jack built, and so on and so forth. Right? A lot of information. A lot of information there, but I think you're getting it. Now, before you leave, let me tell you a little clinical vignette. So my doctor, when I went for my physical, he asked me how you're doing and so on and so forth. And I always tell him, I really like to lose more weight. I'm too heavy. And he said, well, for years, we've been telling people to cut down on fats. We don't tell people that anymore. We tell them now to cut down on sugar, because the American diet has way too much sugar in it. And so I asked him, well, what about complex carbohydrates versus sugars, versus simple sugars? He said, well, as I'm sure you're very well aware, all complex sugars become essentially glucose. He said, so you want to stay away from things that will just dump free glucose into your system. If they get broken down to small sugars gradually, that's fine, because then you'll have this slow release of sugar. It's when you get these big jumps of sugar in your bloodstream, that's what tends to lead to things like type 2 diabetes and things like that. So that is essentially an application of what we're talking about here. Polymers, when you feed them into a system, they have to be broken down. It takes time to break things down. So whereas bacteria cannot get diabetes, this is an application essentially for, I think, people. Because if you eat, for example, a Hershey bar, you're going to get lots of free sugar into your system. If you eat a bagel, now you're going to have starch that's very slowly released into your system, and you won't have the spike in blood sugar, and it'll actually satisfy your hunger for a much longer time. So that's why the dietary advice, which you guys will hand out to your patients, is not so much we need to avoid fats. You need to avoid free sugar so much. And I said to myself, gee, the advice doctors are giving to people is finally catching up with where the research was 10 years ago. So that's my little vignette for you. We will finish metabolism hopefully on Friday. Lab books are in the office. Pick them up. I know some of you won't be here Friday because of core. And those of you on Zoom, thank you very much. And that ends our Zoom session. Have a great day and please get better.